Hey, welcome. Today we are gonna crack the code that makes you, well, you. We're diving deep into genetics, the amazing science that explains everything from why you have your dad's nose to what makes every single living thing on this planet unique. So let's get into it.
So let's start with a question you've probably asked yourself a million times, especially when looking at old family photos.
You know that conversation, right? At every family get-together, someone's pointing out who got what from whom. Well, turns out it's not random at all. The answer is all thanks to a really powerful biological process called heredity.
To put it simply, heredity is just the process of passing traits from parents down to their kids. And the science that studies this whole shebang, how those traits get passed on, why we see so much variety, and all the rules that control it, that is genetics.
Okay, so if traits are being passed down, where are all these instructions actually stored? Well, think of your body like a super complex gadget that comes with its own instruction manual. Let's flip it open and take a look inside.
So, these instructions aren't just floating around. They're all neatly packed into these structures called chromosomes. These are the master blueprints, tucked away safely inside the nucleus of your cells, and they hold every single piece of information needed to build and run you.
For humans, the magic number is 46. Yep, in almost every cell in your body, you've got 46 chromosomes, and they're organized into 23 pairs. And for each pair, you got one chromosome from your mom and the other one from your dad.
Now, not all these pairs do the same thing. The first 22 pairs are called autosomes. They're in charge of most of your physical traits, stuff like your hair color, your height, all that. But it's that final 23rd pair, the sex chromosomes, that has a really specific job, determining your biological sex. It's XX for female and XY for male.
So if the chromosomes are like the chapters of your instruction manual, what are the actual sentences that give the specific commands? Well, for that, we have to zoom in, like way in, to something called a gene.
A gene is just a tiny little section of DNA on a chromosome that codes for one specific trait. But here's where it gets really interesting. For any one gene, you can have different versions. Take the gene for earlobe shape, for example. There's a version for attached lobes and another one for free lobes. These different flavors of a gene are called alleles. Go ahead, try it right now. This little party trick is a perfect example of a trait that's usually controlled by a single gene. The ability to roll your tongue, that's typically a dominant trait. Not being able to, that's recessive.
And this brings us to two words you absolutely have to know: genotype and phenotype. Your genotype is your genetic code, the actual pair of alleles you've got inside your cells. Your phenotype is the physical trait we can actually see. So look at the table. Notice how if you have even one copy of the dominant allele, that big R, you can roll your tongue. That recessive trait, the little r, only gets to show itself if you have two copies.
So how in the world did we figure all of this stuff out? To answer that, we need to go back in time, to a monastery garden in the 19th century, tended by a guy who would become known as the father of genetics himself, Gregor Mendel.
Mendel's genius was all in his super simple, super careful method. He took purebred tall plants and crossed them with purebred dwarf ones. In that first generation, something weird happened. All the new plants were tall. It was like the dwarf trait just vanished. But Mendel didn't stop there. He then took these new tall plants and crossed them with each other. And in the second generation, get this, the dwarf trait made a surprise comeback.
So what was going on? This simple little chart, called a Punnett Square, shows us exactly what happened. Mendel figured out that the first generation plants were all hybrids, carrying both a tall and a dwarf allele. When they were crossed, those alleles separated and recombined, giving us a mix of pure tall, hybrid tall, and pure dwarf plants.
So what did it all add up to? See, Mendel didn't just look, he counted. And when he counted up all the plants in that second generation, he found this incredibly consistent pattern. For every one dwarf plant, there were 3 tall ones. This is the famous 3-to-1 phenotypic ratio.
But if you look at the genes, the story is a little different. The genotypic ratio was actually 1 purebred tall to 2 hybrid talls to 1 purebred dwarf. This 1-to-2-to-1 ratio is a huge deal in genetics, and trust me, it's something you'll definitely want to remember for your exams.
Now, from all these meticulous pea plant experiments, Mendel was able to come up with 3 basic laws of inheritance, principles that are literally the foundation of all modern genetics today. All right, let's break these down real quick. First, the law of dominance. This just explains why all those first-generation plants were tall. The tall allele masked or dominated the dwarf one. Second, the law of segregation. This is how the dwarf trait came back.
The two alleles, the big T and little t, separated during reproduction, which allowed the TT combo to happen again. And finally, the law of independent assortment. Basically, Mendel found that traits like plant height and seed color are inherited totally independently. Getting one doesn't have anything to do with your chances of getting the other.
Okay, we have covered a ton of ground, from chromosomes all the way to pea plants. You ready to see what stuck? Let's try a couple of quick questions.
Okay, think back to the tongue rolling thing. What do we call the trait that you can actually see with your own eyes?
That's it. It's the phenotype. Remember, genotype is the genetic code behind the scenes, and the phenotype is what you see on the outside.
All right, this is a big one, straight out of that Punnett Square. What was the genetic ratio underneath it all?
You got it. The genotypic ratio was 1:2:1, which is what created the 3:1 phenotypic ratio that Mendel actually saw. Knowing the difference between those two is so important.
You know, we've really only scratched the surface. From designing new medicines to editing genes, the ideas that Mendel discovered in his quiet little garden are now at the heart of a huge scientific revolution. So now that you know the basic rules of the game, what's the next big question you think we should be asking about where genetics is headed? If you found this explainer helpful, go ahead and like, share, and subscribe, and don't forget to press the bell icon for more.